Hey guys, back again with another AP Bio review. This one is cells part 2. I'm going to primarily look at the cell membrane, cell transport, cell communication, and cell division. So top 5 topics: the fluid mosaic model, all the parts of the cell membrane; diffusion and osmosis, so transport; cell communication, how cells are able to signal and speak to each other; cell cycle; and mitosis and meiosis, so looking at how cells divide and grow. So first up, fluid mosaic model. This is one of those things that's kind of complicated, but it's really simple once you understand it. So basically, cell membranes are made up of lots and lots of little parts, and those little parts are constantly moving. We know that as the fluid mosaic model. Major components: phospholipids. Cell membranes are made up of a double layer of phospholipids known as the phospholipid bilayer. If you remember, phospholipids have two major parts. They have a hydrophilic head, the glycerol and the phosphate group make it water compatible. And then they have two hydrophobic tails. Nonpolar fatty acid tails are incompatible with water. So those phospholipids line up tail to tail to form a double layer all the way around the cell, and we know that as the phospholipid bilayer. Embedded in that are proteins. You can have embedded proteins, which are actually in the phospholipids, like sticking through the phospholipids. If they go all the way through, we call them transmembrane proteins. You can also have peripheral proteins that are just associated with the membrane, but not actually embedded in it. Those proteins have all kinds of roles, from transport, things like protein channels, pumps, gates. You can have communication in the form of glycoproteins, and then you can have cell-to-cell attachments. So like in our skin, it's very important that the cells can interact through these proteins. And then finally, cholesterol. Cholesterol is a really important molecule because it increases fluidity of the cell membrane. The more cholesterol you have, the more you break up those phospholipids and prevent them from solidifying, especially at cold temperatures. So here's a little picture of the fluid mosaic model. You can see our phospholipids that have both hydrophilic heads and hydrophobic tails. So that polar-nonpolar interaction is really important for the phospholipid bilayer. It keeps the liquid parts of the cell in and the liquid environment out, and that protects the integrity of the cell membrane, of the cell itself. So we've got carbohydrates. They can be stuck to lipids or they can be stuck to proteins. If it's stuck to a protein, we call it a glycoprotein. Literally means sugar protein. These are really, really important for tags for cell communication, like the blood groups are actually descriptions of glycoproteins. And then it can also be stuck to lipids, phospholipids, and we call those glycolipids. I don't have any of those on the diagram here, but they definitely exist. Then we have a transport protein over here. This one happens to be a protein channel. That just means it's open all the time. You can have gated channels, which have to be opened up by certain signaling molecules. You can have carrier molecules, which kind of work like a swinging door. And you can have protein pumps, which are actually powered by an external energy source like ATP. And then you can see this transport protein is moving some molecules across. We'll talk more about that with diffusion and things like that. But in this case, that protein channel is protecting those molecules as they travel through that phospholipid bilayer. They're not coming into contact with the tails as they go from one water-based side to the other water-based side. And then we have cholesterol. Cholesterol, again, very, very important for fluidity. Organisms that have to tolerate extremely low temperatures are going to have a little more cholesterol to help prevent the cell membrane from locking up, from freezing up as it condenses and those molecules come together. All right, 2, diffusion and osmosis. So when you're talking about transport, the most basic type of transport is diffusion. And basically, to understand diffusion, we say molecules move from a high concentration to a low concentration. And that's true. But a better way of thinking about it, a little easier way, is that very crowded molecules will eventually bump and jostle each other until they move to less crowded places. So in biology, we're mostly interested in through membranes. So we say that molecules will travel, if they can, from a high concentration, crowded, to a lower concentration, less crowded, through a membrane. And when they do this, this is 100% passive. The cell doesn't have to invest any energy if a molecule is going to move that way anyway. The only way that the cell really has to get active about it is if that molecule needs to be moved the other way against the concentration gradient from low to high. If a molecule is diffusing, it can either move through directly through the phospholipid bilayer between the phospholipids. If it does that, we call that simple diffusion. Some examples of molecules that can do that are carbon dioxide and oxygen, which is really, really important. That's how we can oxygenate our blood from our lungs, and then we can then send that oxygenated blood to our less oxygenated tissue to give those cells the oxygen they need. If it passes through a transport protein, we call that facilitated diffusion. An example of something that usually has to do that is water. Because water is a polar molecule, it's not going to be able to move past those hydrophobic tails. So by putting some sort of a channel into the cell membrane, in this case we call it an aquaporin, that gives that water molecule that protection as it goes through from high to low. Now, when you diffuse water, we call that osmosis. So the deal with osmosis is that water always moves from a situation where there's more water, hypotonic, to where there's less water, hypertonic. And you can think of it as wet to dry. So the rule of thumb is if a cell is hypertonic, meaning it has less water, more solute, then its environment, which would therefore be hypotonic, more water, less solute, water will move into the cell from hypo to hyper. So think about it. If you put a raisin, which is very hypertonic compared to a glass of water, into that water, then that water from the glass is going to go into that raisin, and that raisin is going to rehydrate. That's osmosis. Okay? Now, if I do the opposite, if I take a cell that's very hypotonic, lots of water, less solute, and I put it into a situation where it's hypertonic, back in the old days, people used to salt meats to keep them from decomposing. And that's because it drew the fluid out of that meat, which then made it so bacteria couldn't grow. When you do that, if you put a hypotonic cell into a hypertonic environment, water always moves hypo to hyper, and the water is going to move out of the cell. You're going to effectively dry it out. So if you're talking about a human body cell, our cells are actually isotonic to the surrounding fluids, our blood and our body fluids. So that's what we want. We want this state of isotonicity. And basically, when cells are isotonic to their environment, water is moving in and out equally, and the cell is at equilibrium. So when you are dehydrated and you go to the hospital, they don't give you pure water into your veins. They give you saline, because saline is the concentration of salt that your cells should have. So your cells, which are dehydrated, will take on water until they reach that ideal salinity of the saline of your blood. So cell communication, cells use signaling molecules to communicate. We call them ligands or signals or whatever you want to say. In human bodies, we talk about hormones, and hormones are usually either protein-based or steroid lipid-based, and they help our cells respond to stimuli and maintain homeostasis based on what is being gathered around us. So some examples of cell signaling within the human body, we use insulin and glucagon. These are protein-based hormones that are secreted from our pancreatic cells, and then they go around through our body and tell our cells how to respond to changes in blood sugar. If you eat something, your blood sugar goes up, and then insulin is sent out around the body to tell your cells to grab that extra sugar and bring it down. Okay. On the flip side, if your blood sugar gets a little too low, you can release glucagon, and that will tell your cells to release sugar and bring that blood sugar back up. Testosterone and estrogen are steroid-based hormones. Because they're lipid-based, they can actually pass through the membrane. They don't require any sort of other receptor, and they go in and they act directly on transcription factors in the DNA. So in human bodies, testosterone and estrogen are able to affect the way that our genes are read, and that can in turn affect our development and a lot of other things. So cell communication has three basic steps. Reception, when a signal molecule attaches to a receptor on or inside our cells. Transduction, when the cell interprets and spreads that signal. And then response, whatever the desired outcome is. So that could be something like reading genes, growing, dividing, secreting other molecules, whatever it is. So again, if you have an insulin receptor on a cell, it receives that insulin. The cell says, hey, there's insulin here. I need to do something about it. And then the response is to bind sugar into starches. Reception, transduction, response. Don't forget that. In the human body and nervous system, electrical charges and neurotransmitters are used to send signals. Those cells receive those, decisions are made, and then there might be some sort of response like a motor function. The immune system is a really, really complicated network of cells and proteins. They interact to respond to any sort of external threat or non-self threat. So if you have some sort of bacteria or a virus roam into your body, your immune system is going to receive information about that, make a decision on what to do, and then enact a response. And then finally, the endocrine system. I mean, this is the hormonal system. It's made up of glands. Those glands secrete hormones that go throughout our body, and they help us maintain homeostasis. So adrenal glands, thyroid gland, those glands tell our body what to do. 4, cell cycle. So most cells grow, copy DNA, and divide over and over and over again until they reach an endpoint, until they reach the end of their lives. Now, there are some cells that don't divide regularly or at all. Heart muscle and some nervous cells are great examples. That's why if you suffer a heart attack or a stroke, you really don't repair that tissue because there's not a lot of regrowth going on. The cell cycle is controlled by chemical signals called cyclins. So once the cell receives those at key points in the cell cycle, it tells it how to proceed. Cells that ignore chemical signals like cyclins can become cancerous. They just kind of divide at will, and that abnormal division can snowball into something much worse. Interphase is defined as the time between divisions. So whenever a cell is not dividing, it's in interphase. And interphase has 3 major parts: G1, growth; S, copying of DNA; and G2, preparation for division. And then once a cell is ready to divide, it goes into M phase. M phase has 2 major parts, usually some sort of DNA division step like mitosis, and then cytokinesis, the division of all the organelles in cytoplasm to fully divide the cells. That cell is then going to go right into the first stage of interphase. First thing it needs to do is start growing again. So how does a cell divide its DNA? There's 2 major choices. You can either do mitosis. Most of our cells do this when they're just trying to make copies. And then meiosis, which is really only used by cells that are going to carry out sexual reproduction. So the production of eggs and sperm is done by meiosis. And basically what happens is while the DNA is copied, those cells are going to be divided twice. And so you're going to end up with half as much DNA as a typical cell. And we know those as gametes. 1/2 cell plus 1/2 cell equals 1 whole cell. That would be the zygote. So cytokinesis is the actual division step. So cytokinesis is not actually a part of mitosis or meiosis. It follows mitosis or meiosis in M phase. And it's when the cell actually pinches and divides. So if you do mitosis and cytokinesis, you get 2 identical cells that match the DNA of the original cell. So you get copies. So in my toe right now, I'm doing mitosis and cytokinesis to grow new skin, hair, bones, muscle, nails. Right? If you do meiosis followed by cytokinesis, you get 4 non-identical cells. Each one has half the DNA of the original cell, and none of them really look alike. So these are the cells that then go on to become ova, or eggs, and sperm. And so by getting a 1/2 cell and having it meet up with another 1/2 cell, that is fertilization. So that's all I got.